Hello, everybody, and welcome to today's webinar, Beyond Borders, Navigating Global Scale-Up for Viral-Based Therapies. My name's Rob Coker, Deputy Editor of The Medicine Maker, and I am delighted to introduce the speakers from Resi Biopharm, who will guide you through this topic via a panel discussion. Jing Zhu, Vice President, Nucleic Acid and Viral Vector Technology at Resi Biopharm, oversees process development activities and technology platform establishment. He has more than 15 years of experience in process and analytical development, chemistry, manufacturing controls, program steering within the pharmaceutical industry. Chief Scientific Officer Kai Lipinski, who gained a wealth of experience in viral vector manufacturing from a variety of roles before joining Resi Biopharm, is central to the establishment of virus process development and manufacturing capabilities, technical developments, and the acquisition of many key clients. Nathaniel Yunt, VP of Business Strategy and Program Management for Resi Biopharm. His leadership has encompassed sterile formulations and fill finish, small molecules, and biologics, drug substance, and drug product, including mRNA, lipid nanoparticles, plasmid DNA, live biotherapeutics, and viral vectors. Raquel Fortunato, the CEO of GenIBet, Resi Biopharm site in Oeiras, Portugal, and a member of the company's board of directors since 2015. Raquel has been working in a CDMO environment for almost 20 years and has strong experience and expertise in the project management of R&D, tech transfer to GMP, and more. Last but not least, I'd like to introduce Jennifer Hain, who will be moderating the discussion. Jennifer is the head of strategic marketing, where she connects ATMP innovators with solutions, with a deep understanding of the market dynamics, customer needs, and regulatory requirements for developing and producing innovative therapies. Jennifer supports commercial strategy and execution for Resi Biopharm. Our experts will be on hand after the discussion to answer any questions you may have, which you can submit at any time throughout the presentation by clicking on the Ask a Question box on your screens. Now let's get started with the main topic. Jennifer, it's over to you.
Thank you, Rob, for the introduction. Before we get started, we have a polling question for the audience, so please take a look and answer that if you could. Okay, so getting into our roundtable discussion, we recognize that there are a few key factors to ensuring success when scaling and transferring processes, either transferring from an external client source or internally between sites. Kai, would you like to give us an overview of a project that had to be transferred quickly from another CDMO to us?
Thank you, Jennifer. Happy to take over and talk about a project where we supported innovative biotech company Iconavir from San Diego to accelerate an oncolytic adenovirus to first-in-human clinical trial within about 1 year. So here was the problem. The customer was really at risk for the clinical supply due to the COVID prioritization in the US, and they urgently needed to find an alternative supplier. After an initial and intensive fit-for-purpose evaluation phase, they decided to go ahead with Resi Biopharm. So while reviewing the critical data and process flow, we realized that some downstream process redesign was necessary to enable robust manufacture of drug substance. What was at the core to rescue the project and deliver on time? Here, 3 key points mentioned: fast process optimization, effective program management, and really a strong partnership with the contract giver. With regard to process development, we in total executed 4 process runs in the scale of 4 to 8 liter. These materials were very important and were used for different purposes, like program reference material, We used it for QC assay validation and also for the first stability studies that had to be aligned with the clinical trial timeline. Effective program management is really very important for your overall success. You need project management on both sides, very strong at the CDMO, but also at the sponsor, to continuously monitor timelines, prioritize topics, and have an escalation process in place should it be required. Then we always emphasize how important the partnership is with the client. If you have very aggressive timelines, both sides have to react within very short timelines, and your project managers must be fully in control of what is going on. Also very important is for us in this project that the client was able to support the tight timelines with their own analytical and process development capabilities. Also, as a sponsor side, you want to have a strong CMC team that supports and have subject matter experts for all the relevant topics like process development, analytics, GMP compliance, and other regulatory compliance. So on the next slide, we just present some of the particular challenges that we were facing to deliver on time. Overall, as already mentioned, we had to redesign the process, particularly the part from bulk harvest to the first chromatography step. And because this is a systemic administration, it will need a very high product titer to administer high doses. So we needed to change the process overall to enable more than 5 E12 viral particles per milliliter in the drug product. There was a significant assay transfer, qualification, and validation scope to be executed within less than 1 year. Also, we had to remanufacture the master viral seed stock, a first production at intermediate scale, and then a 50-liter larger scale production for which we used the drug substance process. Also, because of the restricted timelines, the client wanted us to go from 4 to 8 liter process development glass bioreactor runs directly to the 50-liter single-use bioreactor run without an engineering run. And again, because very high doses are needed for the systemic delivery, the client asked us to further scale up from 50 to 240 liter. And again, this was done to be without an engineering run, but only with some feasibility studies from 8-liter glass bioreactor process run. As mentioned already, the client needed the batch released within 1 year to the clinic. And then stability studies, which came already from representative PD material, had to be aligned well with the timeline for the clinical trial. And last but not least, another request from the client that is currently ongoing is the further scale up from 240 liter to 500 liter. And again, we were asked to do this without an engineering run, but only based on proper depth analysis and a small upstream feasibility study in a small glass bioreactor. So the next 2 slides, I summarize the platform process that we really developed together with the client. This is a fully single-use suspension-based process that we have taken already to 240 liter working volume. And as mentioned before, we are now taking to the 500 liter working volume. This is really a plug and play platform which you can use for basically any adenovirus for clinical manufacture. So as mentioned, it is suspension based. So you saw the cells, you amplify your biomass in shake flask, then you inoculate your first bioreactor, which is either the N-1 bioreactor or already the production bioreactor. Then you scale up the maximum scale, in our case 500 liter, and then you infect and amplify your virus of choice. A couple of days later, you introduce the bulk harvest. This is quite standardized also for adenovirus by adding a detergent, nuclease, and some magnesium chloride. So this comprises the upstream. And the next slide shows you basically the platform for downstream. After the bulk harvest, we start with a train of clarification filters. Then we do a dilution to condition the material for the capture chromatography step. Then there's a second chromatography step to polish the material. And then the final cross-flow filtration step to buffer exchange and over-concentrate the bulk drug substance. And then before we do the sterile filtration, we have the option to add certain excipients before the bulk drug substance is sterile filtered, aliquoted and frozen down to minus 80 degrees Celsius in this case. So on this slide, we give you some information how the product in the end looks like. On the left, you can see a bulk drug substance container. And overall, we achieved a single-use platformable process that we can scale up to 500 liter, and that enables the production of more than 10 to the 12 viral particles per milliliter. This is particularly interesting for clients who want to try systemic application. So all the productions we have done so far are robustly in spec, and we have no out of specification problems really when we produce this virus with our platform. We have a very good low process residuals profile, particularly in relation to host cell DNA. As you can see in the middle picture, this is dynamic light scattering. Here we have a highly monodispersed final product with hardly or not detectable, at least, aggregates in the product. On the right, you see a typical SDS page picture of highly loaded pockets here, which is IPC drug substance is the final material, and ultracentrifuge purified reference material. Again, as you can see in the bulk drug substance container, it's basically virtually particulate-free solution and the typical opalescence that adenovirus shows at high concentration. Also important, we always achieve the FDA target of a total to infectious particle ratio of being under 30. And actually for this virus, we are very good, mostly in the range of 3 to 5. As said already, the host cell DNA profile is very, very good. And even for very high systemic doses, we still stay below the WHO target of less than 10 nanogram per clinical dose. So on the next slide, I just summarize the overall project scope again and the timelines that we have achieved. So the tasks comprised process development, quality control, tech transfer, assay qualification and validation. Then the first master virus feedstock. Then we produced the first drug substance batch at 50 liter and the second drug substance batch at 50 liter. We then proceeded to a second master virus feedstock at 50 liter and then finally to a 240 liter batch. So the most important timeline and critical timeline for the client was to obtain the first drug substance batch at 50 liter scale within 12 months. And this we did achieve in this project. So also with omitting the engineering ones, we cut down the overall standard project time by about 4 to 8 months. And also by scaling up from 50 to 400 liter, although this was not on the critical timeline, again, we did not perform an engineering one. So with omitting 2 engineering ones, we also made sure that we had a significant cost saving for our client. So this is my last slide. It just brings up a few topics, how we try to bring success to our client. First of all, we have very efficient and controlled transfer of all the biological starting materials. Then the Cuxhaven site here has more than 20 years live virus operational experience. And we have particular in-depth oncolytic virus experience covering adenovirus, herpes simplex virus, VSV, arenaviruses, pox viruses, and paramyxoviruses. So all these viruses together, if you go to clinicaltrials.gov data line, you will see that these virus families make up more than 90% of all the ongoing clinical trials. Then we have well-aligned the analytical capabilities between process development and quality control, and also the equipment between process development is well aligned with our GMP equipment. Occasionally, we have to work with large viruses that we cannot sterile filter, and in the past, we have also executed many bacterial projects. Due to this, we are also highly experienced in aseptic processing and validation. As shown on this case study project, we can take clients without engineering runs from process development to GMP to have significant time and money savings. Particularly also important is that we have very good relationships with all the CROs that we use for the testing that we cannot perform in-house, but prefer to outsource. Thank you, Jenny.
Thank you, Kai. I guess it's worth noting that that work was done in our Cuxhaven site, so that just highlights one of the projects done there, so thank you for sharing. Another factor that we hear some customers wrestling with, and Kai, you mentioned it there, is how to deal with a virus that is not able to be filtered. Raquel, would you like to give us an example of how this has been managed in the past?
Sure. Thanks, Jennifer. It's a pleasure to be here and talk about one of the most interesting and challenging processes that we've had to deal with over the past 2 years. And as you mentioned, this was a virus that could not be filtered. Next slide, maybe. And therefore, we were approached by one of our customers. This was already an existing customer with a challenge to manufacture several batches of vaccinia virus, which due to its size could not be sterile filtered. And this was a BSL-2 virus. It was manufactured in adherent HeLa cells with a relatively complex and long manufacturing process that took about 1.5 months per batch. And, well, we had to, given it could not be sterile filtered, it was clear to us that we had to start from cell thaw with an aseptic processing. And this was prior to the implementation of the new Annex 1. But regardless of that, we were already using within our site some of the guidelines that came up within the new Annex 1, as for example, not using water baths, but using incubators for thaw and media eating and all that. So we started by doing, and this was done in parallel with the tech transfer of the process and the scale-up at our site, to do a detailed risk assessment of the entire process. Obviously looking at the process workflow, looking at the materials, equipment, and personnel that would be needed within the process, and looking at all sources of possible contaminations that could be brought in. All this with the aim of getting into a validation protocol that we could execute upon. Next slide, Jennifer, please. As I said, this was a long process with relatively high complexity and many different equipments to deal with. We used a Class A BSC within a Class B background. Next slide, please. We mapped the process in a very detailed way, as said. This was an extracellular virus with a cell train coming from cell thaw into one cell factory via 5 passages and ending in 40 roller bottles. In this case, we've opted to keep the solutions preparation for the downstream processing done in a separate room, in a Class C room, where the solutions were prepared and then sterile filtered into bags. And we did the integrity tests for all those filters. But all the rest of the process was done and treated as an aseptic operation. The DSP included nuclease and protease treatment, clarification, tangential flow filtration with a concentration step followed by diafiltration, and then DS formulation and aliquoting of the bulk drug substance. Next slide, please. Throughout this process, we managed, to give you an idea, approximately 120 liters of different buffers, mediums, and all that. We used the traditional aseptic process qualification approach, where we have used rich media that was collected at the end of the different process operations and then incubated at 22 degrees for 7 days, followed by another 7 days at 32.5 degrees. Given the amount of buffers and therefore during the aseptic processing TSB that we were manipulating, we made sure that we incorporated traceability of the TSB batch that we were using within the validation protocol, so that we could easily find out in case there was an issue with any of the TSB, we could have the process separated into unit operations, and therefore if there was an issue with a batch that we couldn't have seen before with a batch of TSB, we would know, okay, this unit operation still needs requalification, but not the rest of the process. And that has brought flexibility into the approach. The established criteria were the normal ones, so performing 3 successful consecutive runs with no observed contamination, and we had to test all the TSB media used had to pass through the appropriate grow promotion tests. Next slide, please. We have carefully looked into the process and tried as much as possible to broaden the design space for this, because the intent was really, what do we do if during the process we run into problems? Are those within our validated space or not? And therefore, we've really gone through all of this, and we've incorporated, whenever possible, worst case scenarios, such as, for example, simulating 6 passages instead of 5, because we thought, okay, if there is any issue with the cells, then we have the opportunity to do a 6th passage, including a media exchange prior to the infection step, again, to be able to react within the aseptic qualification design space if there was any issue prior to infection. We've tried to manipulate as many containers as possible, so really thinking of everything that could go wrong and making sure that that got into the validation protocol. We looked at the number of operators that we had in the room, and we tried to increase those during the qualification process, of course, maintaining the balance between not having too much people, but making sure that we had room for an extra people if in a situation where that was needed. We've also increased the number of shifts of the normal process, and especially we have increased the number of rotations. So in a normal processing, we would have each shift working for 3 to 4 hours and then rotating at that time. During the aseptic process qualification, we've done shorter shifts to increase the number of rotations and therefore had a worst case scenario. We've also simulated equipment movement to address any possible malfunction of an equipment. So for example, the incubator used for media eating, we have simulated a breakage. We've incorporated into the aseptic processing qualification a media breakage on that equipment. So that would make it needed to exit with the equipment and replace it with the backup equipment. And this was an operation which was included in the validation protocol. We've also simulated the solutions transfer, which I've told you that we've opted to do in a class C, but we've simulated and included the transfer into class B. We've tried to increase, trying not to run completely crazy on it, but finding the right balance between just using the number of sample points that were in the process, but having a little bit more to allow if there was a need for an additional sample point. We've tried to work, so I've not told you this yet, but the volume of DS that we got at the end was approximately 1.4 liters of product. We've tried to work with a higher volume to be in a worst case scenario. We've simulated spills, so both small spills within the BSC and bigger spills within the room. The tests that were done on the TFF assembly, for instance, so the pressure tests and the integrity tests, we have within the validation protocol simulated the maximum duration times. We've done the same thing with old points, so making them within the qualification longer than they would normally be expected to be. We've included an additional TFF pre-rinse only because we knew that could be needed at some point, and then all the ancillary operations, so the preparation of the transfer sets, which were done in-house using autoclaved validated cycles, the transfer of those into the Class B room, so the transport of those stop. We can then readjust and just cut. I've repeated myself here.
Yeah, sure.
Okay, next slide, please. The establishment of a robust contamination strategy was really key here. And therefore, what we've done was that there was always a room fumigation before the process start and after process end. Room cleaning was done with sterile wipes with sporicidal agents. The BSC was cleaned before and after any activities, also with a sporicidal agent. All material was cleaned before entering into the BSC, and there was a particular care taken with the waste. This type of complex processes, they generate a lot of waste as you go through them. It's something that normally people, it's just in there, but when you are dealing with setting up an aseptic processing with this complexity, it's something that you really need to look upon. And then everything around the environmental monitoring, so assuring that all the activities within the grade A and the grade B were monitored and that the process was controlled and the results were under control. Next slide, please. So this is just some of the data from the actual process. So we could see, because in parallel, and I've alluded to that at the beginning, we were doing the tech transfer and scale up of the process, because this was a new process, but also making sure that we implemented the aseptic qualification protocol and validation that would then enable us to manufacture the drug substance. And what we could see in terms of cell viability was that this was really consistent between all runs, so the process was robust. We could see all the time, but this was something that we knew happened, a decrease at P3, which was coincident with when we passed the cells from cell factories to roller bottles. And next slide, please. We could also see this consistent behavior when we look at the DS, at the downstream data, and where we could see a decrease on the content of impurities. And we have at a certain point, and you can see that for the last 3 runs, the total BSA at the end product is lower than for the first ones, because of the pre-work that we had done on the validation of the aseptic process. We've done this in such a way that we could incorporate the use of a TFF membrane with a larger pore that was needed for product quality at some point, without needing to fully requalify the aseptic processing, but just doing one additional run with this. So next slide, please. So Overall, we have done 6 APQ runs and 7 DS runs in a time frame of about 2.5 years. So we did the first 3 qualification runs, and then every 6 months the process had to be requalified, and it was done through 1 requalification. All batches went very well with 100% batch release, and this was really key to enable the success of the client, because they were in need of this material for the clinical trials that they were running, and we were able to deliver that in a consistent and on-time way. As Kai has highlighted before on his presentation, we think that a close project management and really a collaborative approach with our customers, trying to together anticipate the risks and working on mitigating them, has really been key and is key and instrumental on running all processes at our facilities. Thank you.
Great. So thank you, Raquel. I think it's safe to say that we have a number of highly qualified viral experts and have experience working with challenging viruses under high pressure situations. Jing, would you like to talk about what's next for Resi Biopharm in viral-based therapies?
Sure. Thank you, Jennifer. I think one of the major challenges to work with CDMO is the limited flexibility. That limitation could be operation resource in terms of the facility, personnel, or the limitation could be lack of selection of different technology platform. So at the Resi Biopharm, we don't want simply be the service provider with that limited scope. So we would like to partner with our clients to offering that flexibility. So we go through the journey together with the client. So in the next couple of slides, I would like to show some case study to demonstrate how our flexibility can benefit our clients. So the next slide. So currently we have 3 facilities across both the US and Europe. While all the 3 facilities can offer the service for viral vector, each of the facility actually have its own technical core team who can initiate and lead the efforts. We also have the PD operation teams also cross-trained through all the facilities. For instance, the upstream teams was cataloged by different cell culture, and the downstream team focused on different unit operation, including filtration and chromatography. Based on the priority of the customer project, this downstream team and upstream team can be flexible and shifting around to meet the different needs to address the client's needs there. And the next slide, as we mentioned earlier, for most of the CDMOs, they have their own technology platform. So the offering will be very limited outside of their comfort zone. At Resi Biopharm, we do have our platform for both AAV and Lenti, while we're also more than happy to take any new process if that's preferred by our clients. In order to provide the flexibilities, we are actively exploring and also adopting different technologies in the market. So here is the example for the AAV transfection system. So we all know the transfection region is very critical in this system. So in-house, we have a library of different transfection region. We also optimize the conditions for each of the region. So as you see in the left graph, that's the final titer yield after fermentation of each of the region. So all these regions within our optimization showing pretty decent titer, with 2 of them already outstanding. And to further pushing them up, we also introduced the enhancer, which is very new technology just be introduced in the market. So in our hands, this enhancers really boost all the performance for both AAV8 and also AAV6. Particularly for AAV8, we have seen almost a 3-fold increase of the titer, which as high as 10 to the 15th VG per liter, and also almost 40% full versus empty ratios just after the fermentation, not even go through the purification yet. So we also would like to have this excitement to see how we can leverage that for our incoming client programs too. So similar approaches in the next slides is the downstream side. And we have in-house library of different filter media and also purification media in order to meet different needs. So here is the example of comparison of the two affinity resins which come from the internal database. So first thing we did is the optimization of the operation condition for each of the resin, and we present two best conditions for each of them. So both of the resin showing pretty decent binding capacity, but one of them really showing good recoveries compared to the other. In terms of impurity profile, both of them are very similar. However, we do notify one significant difference during their elution conditions in terms of the pH and the conductivities. So in that case, if we do have an AAV construct, which is very sensitive or very unstable to different environment, then the selection of these resins are very critical to impact their final performance. In addition, we also carry out the base stability test. So in a way, we can offer the recycle of the resin if that's favored by the clients. So the next slide is analytics. So we also believe analytics is equally important as the process. So it's important to have the full control of the testing. So we have in-house capacity for all the testing panels, except some of the safety testing. Not only for that, we also offer more than one of the testing methods for some of the critical assay. So in a way, we can have multiple assay methods to verify the development results to give us a high confidence. So one example here is the full versus empty capsid ratio. At PD, we have the standard for a quick dirty runs, and then we have the capillary electrophoresis with more QC-friendly, more accurate tests. We're also in the process of developing the HPLC-based method. So we have triple methods to fully verify the results. And again, our philosophy here is we would like to provide you the flexibilities, either by leveraging our own analytical platform, or we take tech transfer of the assay developed at the client sites, or we develop the assay together with the clients. Back to you, Jennifer.
Thank you, Gene. So Nathaniel, I know you look at our whole network, not only our network of sites, but our network of resources and our supply chain as well. Can you give us an idea of how this built-in flexibility serves our clients best and ensures that capacity is available where and when it's needed?
Yeah, thanks, Jennifer. And you've heard a lot from Kai, Raquel, and Jing about success stories at the local site level here. But as Jennifer mentions, from a network perspective, how we package that for you as a customer is really important to us. So I've broken this into 5 different ways to look at it. One, from a CMC considerations perspective, applying the scientific expertise that you've heard from the team here, commercial innovation for ATMPs and how we can change that and be creative together, controlling the cost of cell and gene therapies, which is ever more important for this space, and obviously being globally present for your local needs and understanding that footprint and how it serves you best at each location. So on the CMC side, you want a CDMO partner that can support and inform your CMC strategy. You want them to improve and enhance your engagement with the regulators. And you want them to have a level of GMP that does not toggle with the type of product or the phase of the product that you're working on. So from a CMC perspective, what you're doing in phase 1 it's for phase 1 clinical, but you're setting the foundation for your phase 3 and your commercialization in the future. So what we're doing here to support this is, one, being a strategic partner for you and offering advice and counsel if that expertise is not in-house for you, if you're a smaller organization. We bring our CMC learnings from multiple modalities to how they could apply in this viral application. And the way that we generate our documents for you is not just what's convenient for us. We've designed all of our documents to directly support you generating your module 3. because at the end of the day, we don't want you to have to comb through all of our records to try to figure out what the process is. We want to give you a very clear description and good documentation of the process that we've developed for you or that we're executing for you at our sites. At our US sites, we have DMFs that you can cross-reference. And ultimately, we're even willing to write your Module 3 for you for your IND if that's something that would be helpful for you. Because we work with a lot of different customers too, we get different types of engagement with the FDA and other regulatory agencies. So we can help to inform the types of questions that you can ask as you go in for your pre-IND meetings or post-review, so that you get the most value out of the answers to those questions from the agency. And then finally here, when we say phase appropriate doesn't mean non-GMP. We can toggle our CMC and documentation approaches with you to make your phase 1 less burdensome as you go in with a fairly new product, but we do not toggle the level of GMP that goes along with that. For us, GMP is GMP, and that's not going to be tailored down or discounted just based on a phase 1 program. So you've heard from a few of our experts here. There's a lot of specialization that goes on at our different facilities and our different teams. And what we see is that often high specialization doesn't come with a broad level of support. But what you find with Resi Biopharm is that you have that specialist strength at 2 scales. One, at the Resi Biopharm level from our business unit, you have a real depth of technical expertise, a lot of robust process development experience around vectors, oncolytic viruses, hard to filter viruses. But along with that, you have the support of broader Resi-Pharm, which has a great deal of experience around commercializations of lots of different types of products, and a strong global PAI readiness support team that can be implemented and support each of our facilities worldwide. So taking these 2 scales of strength, we think we can offer a really strong powerhouse to bring these advanced therapies to market that you might not otherwise get with one or the other in terms of specialization. But at the end of the day, to us, execution is the strategy, whether it's how we support you from a highly technical standpoint or from a broader support. Strong execution is the lowest common denominator for us to deliver you a quality product. So continuing with that global theme, Resi-Pharm broadly has support across North America, Africa, Europe, and Asia, providing you a really wide global footprint to go along with the international footprint that Resi Biopharm offers as well. Next slide. So when we talk about creative commercialization, these being advanced therapies and new types of products, there's not a lot of commercial history to go off of. So we often get questions about commercial facility when customers come to us. And to a certain extent, it might be the wrong question. And you can look at this from 2 different perspectives here. One from a quantitative controls infrastructure and one from a cultural infrastructure. Obviously, when you're picking a CDMO partner, you're not wanting to tech transfer multiple times throughout your development process. So you want to find someone who can commercialize your product. Important things here: equipment qualification strategies and management of those pieces of equipment that deliver the ultimate drug substance or drug product at the end of the day. Strong quality systems and process management. So not just systems that manage your processes from a deviation perspective, but also continuous improvement systems that provide you more and greater efficiency as your product develops over time with us. And you need a strong supply chain to ensure continued supply of this product as you commercialize it. But the other side of this that might be slightly less tangible is the culture and drive of your CDMO partner. And this is where commitment to delivery, a bent towards customer service, and patient impact being the driver for delivery is exceptionally important. And quality is woven through all of this because when patient impact is the driver for delivery, it is woven into all of the approach when it comes to execution. down to the operator level, when they know what they are producing really matters in the lives of patients in the clinic and hopefully ultimately as a commercial product. But delivery and consistent service is something that can't be undervalued when you're looking for a commercial partner in a CDMO. But because these are all new therapeutics, the agencies are looking for ownership and a robust understanding with CDMOs and partners, because this level of engagement is setting the groundwork for future approvals. So the way we go about this is exceptionally important. So if you follow our industry news, the drumbeat of CBER is that the cost of cell and gene therapy development and manufacturing has to come down to ensure long-term accessibility for patients in the future of the modalities. So part of this is what you've heard, with Kai talking about the solution with Iconovir, smart platforming with partners, developing processes that are robust, that have high yields and high purities, so that it's not just a solution for this product, but for the customer's next product in their pipeline as well. You heard a little bit from Jing about having an efficient design of experiment that doesn't waste development activities. You have to make every experiment count when you're developing these types of viral products. And then you need a CDMO partner that's going to be in the trenches with you and have an appropriate balance of risk sharing and risk mitigation. You heard from Raquel that it's not a straightforward task to do an aseptic qualification of a virus that can't be filtered. But when the team in Portugal put their heads down and figured out the appropriate strategy, they're able to produce something that's exceptionally reliable and a really creative solution. And then finally, time is money. We understand this from your perspective, that reducing the timeline from kicking off with us and getting IND enabling data and getting into the clinic might mean another year of time for you to continue innovating and finding new products to bring to the market. So everything that we're doing here is to challenge these conventional timelines and provide you with a fast and reliable solution that we can deliver with a high level of integrity and credibility, ultimately to make this a long-term solution for patients. And along with our global strength here, we want to be agile local partners. So you've hopefully gotten a sense for that agile, creative, flexible, solution-oriented approach that Raquel and Kai and Jing have outlined for us. But each of our sites is meant to serve a strategic benefit to our customers. And we think that our presence in the US and Europe further provides strong support from a regulatory perspective, depending on where you might want to execute your trial or ultimately do commercial distribution. So appreciate that you've taken the time to learn a little bit more about our solutions here. And we would love to be your next creative partner in bringing your viral product to market.
All right, well, thank you, Nathaniel, and sincere thank you to all of our speakers, some of my favorite people to work with, and it's been a great time spent with you to hear a little bit more about your areas of expertise. Before we get into the Q&A, we have a final poll question for our audience, so please take a look, and if you could answer that for us here, then we will get into the Q&A. Thank you very much.
Many thanks, Jennifer, and thank you to all of our speakers here today. We are ready to start the Q&A session. Please submit any questions you have using the box on your screens, and we will try to get through as many as we can. The first question is, what factors should I consider when deciding between the European and US sites for clinical or commercial manufacture? Raquel?
Yes, thank you, Rob, for that. I think it really shouldn't matter for you as a client what is the site that you are choosing for and whether it is in the US or in Europe. The way that we are setting up the unit for biologics, it's really making sure that we have a unified quality system and a unified ways of working that really complies with both the GMPs in Europe and the requests from the European Medicine Agency and those from the FDA. And for that, really the choice for the location will be based on what, from a technical perspective, may fit better, from a technical and a timeline perspective, what will fit better the customer and the project needs at that time.
Thank you. Okay, we have another question. Every CDMO says they are flexible and end-to-end. Is Resi Biopharm really a flexible CDMO with an end-to-end offering? Nathaniel, I think that's for you.
Yeah, absolutely. So I fully expect that more and more CDMOs will enter the language of an end-to-end service provider. So that I don't expect will be a long-term differentiator for you as you look at different CDMO partners. But the way to approach that then is to really focus on delivery. Similar to what I mentioned previously in the slides is finding a partner that is exceptionally razor-focused on delivering for you is the key piece. You can have the best technology and an end-to-end offering for a customer at the end of the day. If you're failing batches continuously or you can't meet an accelerated timeline, that end-to-end offering is not so useful to you. So yes, I would say Resi Biopharm is imminently flexible. We want to deliver for you, and we want to build our business based off of delivering high-quality clinical and commercial material for you. And when it comes to what flexible really means, it goes even beyond that mindset into the infrastructure. And what we've built with our quality systems is really designed to flex to your needs, not from a GMP perspective, but from a CMC and a documentation support framework. So from the attitude and the culture down to the systems that are used along with that drive, there's a high degree of flexibility that we can offer on both fronts.
Okay, thank you, Nathaniel. How comfortable is Resi Biopharm in working with unusual process steps, viral vectors, or customer requests? Jing?
I would say we never shy away from any challenges or new process. Two things I think give us the confidence for that statement. First, we have a very strong PD teams, pretty good expertise within the house in terms of process, analytics, tech transfers to cover from small scale to final GMP readiness. And second thing, I think also very critical, our philosophy is we believe certainly not one fit for all. For the best interest of every client, we prefer to do the customized process development so we can serve the best interest for their needs. Yeah.
Thank you, Jing. I think we have time for one more question just now. How will I know what is going on with my project once I hand it over to you? Kai?
Well, as mentioned already in my presentation, project management is key to the success of the overall project, and we live with 100% transparency with our clients. It doesn't help to hide anything and to be non-transparent, because GMP also means that everything is transparent and accessible and visible. So this is clearly our approach, and hiding anything is not something that helps, and it always hits you later in the project. So this is what you can expect from us, a trustable and fully transparent partner within your project, from beginning to the end.
And that is the end. Thank you very much to each member of our panel for those answers. Thanks. That's all we have time for today. If your question hasn't been answered live here, don't worry. We will pass all the questions over to our speakers so that they can follow up via email. And if you wish to revisit the topics covered today, this webinar will be available on demand in the next couple of days. Thank you again to all of our experts from Resi Biopharm for sharing their expertise, and thank you to all of you for attending. I look forward to seeing you again at future webinars.
Thank you.
Bye.